Fine. A very good morning, all of you. So, welcome to our day 16 of our 21-day challenge. So, we're nearing our target. So, before we start, how are you guys doing? Did you have a good night's sleep? So, as days are progressing, we tend to sleep early and tend to wake up early without any external assistance, isn't it? So I hope you guys are all ready. So let's start with our presentation. And if you have any issues with streaming audio or video, do let me know in the comment section, right? So without much delay, let's go ahead with our presentation. Again, a general discussion followed by subject discussion. Fine. So day 16 of our 21-day challenge, we'll start with the following general topic. Often we are subjected to criticism. If it is from external side, that's fine. However, unfortunately, in most of the situations, we resort to harsh self-criticism. So, is that good? Does it help us improve or accomplish our goals or targets? So let's see. So forgive yourself as well as others. Yeah, thank you, Dr. Nidhi, for the feedback. So not only do we incur emotional wounds from others, most of us inflict them upon ourselves. In fact, if we compare these emotional wounds from others to self, ours will be a major portion. We beat ourselves over the head with self condemnation, remorse, and regret. We beat ourselves down with self doubt. We cut ourselves up with excessive guilt. Remorse and regret are attempts to emotionally live in the past. Excessive guilt is an attempt to make right in the past something we did wrong or thought of as wrong in the past. This is the most significant point. Remorse and regret are attempts to emotionally live in the past. What's the point? Emotions are used correctly and appropriately when they help us to respond or react appropriately to some reality in present environment. Since we cannot live in the past, we cannot appropriately react emotionally to the past. The past can be simply written off, closed, forgotten, insofar as our emotional reactions are concerned. We do not need to take an emotional position one way or the other regarding detours that might have taken us off course in the past. The important thing is our present direction and our present goal, which is going to decide our future. So we need to recognize our own errors as mistakes. Otherwise, we could not correct course. Steering or guidance would be impossible, but it is futile and fatal to hate or condemn ourselves for our mistakes, isn't it? So, with these introductory general comments, let's move on to our subject discussion. So, there's no point in living in past, and the best thing and the smart thing would be to focus on what we have to do now. Understanding the mistakes which were committed previously helps us or guides us to be better versions of ourselves. But hating ourselves just because we have done something in the past, right? Getting emotionally strained is in no way going to enhance our present or, for that matter, our future. So, now let's move on to our subject discussion. So today it's an amalgamation of biochemistry with microbiology, or for that matter, in specific living organisms. So previously, if you remember, we have seen about respiration, various cycles, how they are linked in a process of catabolism or to yield energy. So today we'll go to even further basic topic, that is metabolism. What is metabolism all about? What is its significance and relevance in living organisms, right? So in the process, I'll be highlighting a few important points. As you can see in yellow, you can make a note of them, or you can go through them again and again after the session concludes during revision time as per your convenience. So metabolism, of course, can be considered fundamental to life. So any reaction that happens, which is essential for survival, there are infinite number of reactions happening. So metabolism is sum total of chemical changes that occur in living organisms and which are fundamental to life. All prokaryotic and eukaryotic cells are metabolically active with an exception of virus. Even the virus needs a host which is metabolically active, which is very interesting. So we're most worried about viruses, especially during these pandemic times. And look at the irony: there is no active metabolism in virus, and still it's one of the most dreaded things to humanity. So all prokaryotic and eukaryotic cells are metabolically active. The sole exception is viruses, but even viruses require a metabolically active host for their replication. So when viruses do not have metabolism on their own, can we call them living or should we call them non-living? Okay, so metabolism involves the use of compounds. So there are two ways of using these nutrients or compounds, that is anabolism or catabolism. So nutrients from the environment are used in two ways by microorganisms. They can be building blocks of various components of microorganism, assimilation or anabolism, or nutrients can be degraded to yield energy, that is opposite of assimilation, dissimilation or catabolism. There are also amphibolic biochemical pathways. And the best example is tricarboxylic acid cycle, TCA cycle. So some so-called amphibolic biochemical pathways can serve both purposes of anabolism as well as catabolism. The continual process of breakdown and resynthesis are in a balance that is referred to as turnover. And metabolism is an open system. It's not closed, it's an open system where various products can enter and exit. So that is, there are constant inputs and outputs. A chain of metabolic reactions is said to be in a steady state when the concentration of all intermediates remain constant despite the net flow of material through the system. That means the concentration of intermediates remains constant while the product is formed at the expense of substrate. So we have substrate. We'll look into that in the form of an equation a bit later. So you need to understand the fact that we say that these metabolic reactions are constant when the intermediates, the concentration of intermediates remains constant, not that of substrate, not that of product. Okay, so primary metabolism, secondary metabolism, where primary metabolism comprises those metabolic processes that are basically similar in all living cells and are necessary for cellular maintenance and survival, which we come across in the form of various reactions or cycles in biochemistry. They include fundamental processes of growth, that is synthesis of biopolymers, macromolecular structure of cells, organelles, energy production like glycolysis or TCA, and turnover of cell constituents. Whereas secondary metabolism refers to production of substances such as bile pigments from porphyrins in humans, which only occur in certain eukaryotic tissues and are distinct from primary metabolic pathways, which we come across in biochemistry. In our curriculum are mostly primary metabolic pathways. Metabolic control process that occurs inside cell include regulation of gene expression and metabolic feedback or feed forward process. So this is regarding positive and negative feedback, which is controlled by genes. The triggers of differential gene expression may be physical, chemical, or environmental. Differential gene expression is responsible for regulation at molecular level of differentiation and development, as well as maintenance of numerous cellular housekeeping reactions, which are essential for day-to-day functioning of microorganisms. In many metabolic pathways, the metabolites, that is, substances produced or consumed by metabolism themselves, can act as signals in control of their own breakdown and synthesis. That is, either a positive or a negative feedback, either through positive or negative feedback. Feedback control can be negative or positive. Negative feedback results in inhibition by an end product of the activity or synthesis of an enzyme or certain enzymes in chain reaction. The inhibition of synthesis of enzymes is called enzyme repression. So, inhibition of activity of an enzyme by an end product is an allosteric effect. And this type of feedback control is well known in many metabolic pathways. Allosteric effect is nothing but a ligand binding to a specific site on a protein molecule, such as an enzyme, so that there is a configurational change at other sites of this protein molecule or enzyme, right? So many enzymes are allosteric proteins which can be inhibited this in this particular way. So in positive feedback, so that's negative feedback. In positive feedback, then product activates an enzyme responsible for its own prediction for perpetuation of the cycle. So this is a basic overview for us to understand how or why we call these metabolic reactions or cycles as being open, and what do you mean by constant, or rather we can say stable reaction. Many reactions in metabolism are cyclic or metabolic cycle. That's the reason why we call it a cycle, because the end product, uh, one of the end products, again sustains the reaction, hence cyclic. A metabolic cycle is a catalytic series of reactions in which the product of one of the involving two molecules is regenerated. As you can see, A plus B gives rise to C plus A. So A is being regenerated and again being used in the cycle. So thus A acts catalytically and is required only in small amounts, and A can be regarded as carrier of B. The catalytic function of A and other members of metabolic cycle ensure economic conversion of B to C. B is the substrate of metabolic cycle and C is the product. If the intermediates are withdrawn from the metabolic cycle for biosynthesis, the stationary concentrations of metabolic cycle intermediates must be maintained by synthesis. And replenishment of depleted metabolic cycle intermediates is called anaplerosis. So this is something which I wanted to highlight. So do make a note of this. So replenishment of depleted metabolic cycle intermediate. So this replenishment happens, and when intermediates remain constant, we consider that cycle or reaction is stable. So replenishment of depleted metabolic cycle intermediates is called anaplerosis. Anaplerosis may be served through single reaction or a chain of reactions leading to supply of these intermediates. And also we have some examples regarding the same. So anaplerosis may be served by a single reaction which converts a common metabolite into an intermediate of metabolic cycle. An example of this is pyruvate to oxaloacetate reaction in TCA cycle, which we are all familiar with. Alternatively, it may involve a metabolic sequence of reactions called anaplerotic sequence. An example of this type is glycerate pathway, which provides PEP, phosphoenol pyruvate, for anaplerosis of tricarboxylic acid cycle. So you can make a note of these examples and also what anaplerosis is all about. Now, penultimate points: prokaryotes and eukaryotes. So prokaryotes exhibit a great diversity of metabolic options, even in a single organism. For example, E. coli can produce energy by respiration or a fermentation. Respiration can be under either aerobic or anaerobic conditions. When we say aerobic, as we discussed in one of the previous 5 AM sessions, where the oxygen is a final electron acceptor in case of aerobic respiration. Anaerobic, it's other than oxygen. So compounds like lactose or glucose can be used as the only source of carbon. Other bacteria have other metabolic capabilities, including the use of sunlight for energy. So this is basically how various prokaryotes survive through various means and mechanisms. And some of these mechanisms are also utilized by eukaryotic cells. In addition, prokaryotes have a number of energy generating mechanisms that do not exist in eukaryotic cells. Prokaryotic fermentation can be uniquely done via phosphoketolase and enter Diodarov pathways. Anaerobic respiration is unique to prokaryotes, as is the use of inorganic compounds as energy sources or as carbon sources during bacterial photosynthesis. Archaebacteria possess metabolic pathways that use hydrogen as energy source with production of methane and a non-photosynthetic metabolism that can convert light energy into chemical energy. So various examples giving us a glance on how metabolism happens in different types of prokaryotes. And Bacteria, metabolic process are coupled by synthesis of ATP, the principal fuel source of cell, through a series of membrane-bound proteins that constitute ETC, which were seen previously, electron transport chain, electron transport system. The movement of protons from inside to outside of membrane during the operation of electron transport system can be used to drive many processes in the bacteria, such as movement of flagella used to power bacterium along, and synthesis of ATP in the process called oxidative phosphorylation. And before we conclude, the fermentative metabolism that is unique to some bacteria is evolutionary ancient. This is consistent with early appearance of bacteria on Earth related to eukaryotic organisms. But bacteria can also ferment sugars in the same way that bring yeast up ferment sugars to produce ethanol and carbon dioxide. This fermentation via the so-called Embeden-Amerov pathway can lead to different end products in bacteria such as lactic acid, example lactobacillus, mixture of acids like Enterobacteriaceae, butanediol in case of Klebsiella, and propionic acid as end product in case of Propionibacterium. So this is an overview of metabolism, especially in prokaryotes and also in bacteria. In humans, more or less, the primary metabolic pathways remain the same. However, due to various evolutionary changes, we even have secondary metabolism and so many cycles happening at cellular level, which keep us going. So this overview of metabolisms, understanding the significance of various cycles and how they are essential for thriving life, gives us a broad view on various metabolisms, and also uh, to have a summary of all that we have discussed so far. Initially, we have seen about harsh self-criticism and how it damages us, especially emotionally and of course physically as a consequence. So realizing our mistakes is significant, but not just criticizing ourselves for something that has already happened. In fact, it serves no purpose. And the best thing, the smart thing would be to focus on what we have to do now, which is ultimately going to affect our future, not the things that have happened in the past. And also, you've seen metabolism, an overview of metabolism, and also you've seen certain terms and various types of mechanisms, what anaplerosis is all about, and how metabolism in a fundamental way is affecting all life forms on this planet. So, with this, let's conclude our 5 a.m. session. So, it's been amazing interacting with you, sharing information, and then most importantly, the basic thing of waking up early in the morning itself is very exciting. I hope you're experiencing the same, and your feedback is always welcome. So it's all about working hard, working consistently, and most importantly, believing in ourselves. So let's conclude and wish you all a fantastic day ahead. Love you all. And if you're feeling sleepy, don't try to sleep immediately. Uh, Work on your priorities, and you can have a power nap at around 9:30 or 10, and make sure that you're sleeping early every day if you want yourself to be waking up early. If you want to see yourself waking up early and focusing on various priorities, then the basic thing is to sleep early. And also the kind of food which we take plays a major role. We'll discuss more of these facts along with literature review and literature references in the coming sessions. And I'll be seeing you again tomorrow in 5 a.m. club. So wish you all a great day ahead. Love you.